Well, I think we're ready to go here. Thanks, everyone. Thanks for being here. I'll give you all 7 more seconds to stop talking. Well, thanks, Oyster South. Thanks, Bill and Beth and the Oyster South team. It's great to be here. We're going to talk to you about sudden unusual mortality syndrome, SUMS. There are 4 of us panelists. Amazing. I can't do that. There are 4 of us panelists, but we had 1 extra panelist in my graduate student, Colin Elmers. I just told him to get out of here. He's flying back. We have a generational snowstorm hitting the Carolina coast, and he managed to find a flight that's leaving right now. I think I'm stuck in Texas for the next week, maybe, which I'm kind of excited about. So there are 4 of us talking about some of the things happening in our labs and in our states related to unusual mortality. And this is something that's really normal for scientists to get together and talk. We go to conferences all the time, we share information. I think what's really valuable and just awesome about doing this in Oyster South in general is that we as scientists get to share this information with much broader communities. And kind of Oyster South is kind of turning into, well, one, it's the best conference, but it's kind of turning into a forum unlike, I don't know anything else like it, where there's this kind of direct extension. Everyone up here, yeah, everyone up here, you're gonna hear some talks by a bunch of professors right now. But we're all in really interesting professorial kinds of positions where we teach and do research, but everyone here has a really large extension component in the work they do, and that's really unique in academia. I don't think I would enjoy any other position where the work is really applicable, and it's really the relationships that have really built up the kind of work that I do in my lab. Well, one, I'll start. I'm Tal Ben-Horin. I'm a professor at North Carolina State University. I'm at the College of Veterinary Medicine, but I'm not a veterinarian. I'm a pathologist. We have Andrea Tarnecki here, who's at Auburn University, so runs a shellfish research and extension program. Ryan Carnegie is another shellfish pathologist at the Virginia Institute of Marine Sciences, and that's probably the longest serving ever shellfish pathology program. And then you get Bill. Bill was a last minute addition as of about 10 minutes ago. I asked him to come up here, but the reason I asked him to come up here is because Bill threw a slide at us yesterday or the day before that we stuck into Colin's talk, so now I'm gonna make Bill do that. So, I'm gonna start by giving you a broad overview of what I've been seeing, what my lab has been seeing in North Carolina. And in general, I'm a pathologist, so we're going to talk about pathology a little bit. I promise I'll make it engaging. And then I'll pass it off to both Andrea and Ryan and Bill as well. And so to start, I mentioned I'm a pathologist. I run an active field and research program in North Carolina. In North Carolina for a while, we had a few years in a row where oyster mortality was known as Mother's Day mortality. One of the things that I do as a pathologist or as a marine ecologist is we go out, we get a bunch of oysters, we put them at a site, and we just wait for them to die. And this is what we're looking at here is one of our deployments where this is a site where the grower that we were working with had near 80–90% mortality year after year. This wasn't his only lease. And so this wasn't his livelihood, but we just knew something was going on in the site. And it's, for me, starting a research lab, this is really valuable where I can have a site where I knew whatever's happening related to SUMS is happening right here. And so what we're looking at here is right around Mother's Day, all our oysters went away. And what's really was really striking about this, for many years in a row, we saw year after year mortality happening on Mother's Day. These are data from 2022. Here are the data from 2023. It's like to the day. Another thing that's really interesting about these patterns is, as the state shellfish pathologist, I talk to growers across the state, not just in the places where I have oysters deployed. And what we see, my phone would start ringing kind of like early May. Growers across the entire state, separated by hundreds of kilometers, are seeing the exact same thing happen, right around the exact same time. I showed data from calling it Mother's Day mortality. Now that we've been doing it for a bunch of years, it isn't just Mother's Day. It's kind of the whole, I'm going to show you some data. It's anywhere from May, June, all the way through November. And Andrea's going to show you some data from the Gulf where that also seems to be the case. I mentioned I'm a pathologist. One thing that we're able to identify, since we're doing these deployments where we put oysters out and then we kind of have a pretty good hunch that mortality is going to happen at these sites. And so we're able to set up sentinel studies where we're sampling oysters ahead of the mortality, just seeing what's going on. And one of the first things that we identified was just a lot of inflammation. And that's what we're looking at here. This is kind of a really low magnification look at a cross-section of an oyster that was collected about 3 weeks before any mortality was observed on the farm. And this sample was taken from 2022, one of those first curves that I showed you. And what you're looking at here, I'm not going to point, I don't want you to point out the individual detail, but if you look at all those little circles throughout the periphery of that oyster tissue, you just see a lot of that like blue, that blue, kind of dark blue purple color. What those are oysters' inflammatory cells, hemocytes, that are aggregating in a lot of areas through a lot of the tissue. And what we're seeing here is like wide-scale inflammation in the digestive tissues. This would be like an oyster with irritable bowel syndrome, or Crohn's disease, or celiac disease would be the human analog to this. We're not seeing any of the known pathogens, the things that we know cause mortality in oysters. What we just see is this kind of unexplained, multifocal, or sorry for the jargon, just broad inflammation happening everywhere. And it's not just the digestive tissues that just got cut off. It's in the mantle, and it's in the gills. And I know Ryan's seen inflammation, quite a bit of inflammation in gill samples. We could zoom in a little bit, and the top image is that same exact oyster, but just zoomed in. And what you're looking at are digestive tubules with all those inflammatory cells aggregating in. What's really interesting here is the digestive tubules themselves kind of look okay. They look fine. There's not a lot of pathology. If we look at the weeks after this, we start to see a lot of pathology in the digestive tubules. And what these images are showing you, the images on the right, what they're showing you is, as a pathologist, you can do a lot of fun things. One of the things we did here is known as tunnel staining. This is looking for breaks in DNA, which are indicative of cell death, apoptosis. And so these are just complementary images to what's on the left. So with all that inflammation, there isn't a lot of cell death early on, but as time progresses afterwards, we're starting to see the digestive system itself start to die. So ultimately, the cause of death here for the oysters to die is probably related to starvation or lack of nutrient assimilation, because these are the sites of nutrient assimilation. Essentially, these oysters are starving to death. And here's one of our deployments, and this is just a time series, and all this is showing is that as we move through time, we start seeing more and more of that pathology, even before any mortality is starting to occur. And probably about 2 to 3 weeks after that initial pathology is observed is when we start to see oysters die. I've been really fortunate in my lab. I work really closely with, as being at a university, I have a lot of wonderful graduate students and postdocs who have been really interested in this. One of our postdocs, Dr. Stephanie Smith, came to us where they worked with, did a lot of work on squid symbioses, Vibrio bacteria associated with squid, and was wanting to do just characterize some of the microbes that are associated with oysters, both leading into and after mortality. And what Steph has been able to identify is really, really neat. What Steph has found is that leading into the mortality event, when oysters are healthy, there's a suite of resident bacteria. Handful of resident bacteria. One of them that always seems to be there is known as Vibrio mediterranei. And as we move into further and progress into mortality, Vibrio mediterranei goes away. There's a lot of shuffling in those communities. And what it gets replaced by is a handful of species. The species isn't always the same. It's just the community seems to shift. Sometimes it's Vibrio alginolyticus, sometimes it's Vibrio agravarians, sometimes it's Vibrio harveyi. The key take-home message is across all these mortality events that we've observed is that we're constantly seeing these, one, Vibrio mediterranei goes away and a bunch of other species go away, and it gets replaced by other taxa. What Steph's been able to do is look at the genes, sequence the whole genomes, and ask questions about functionally, what's the difference between Vibrio mediterranei and some of these other species? And we've got lists and lists of genes that are associated with those resident, maybe symbiotic Vibrio mediterranei, and then genes associated with everything that shifts to replace it. And what we see is just kind of shifting in the function of what those other Vibrios are doing, the Vibrio harveyi or the Vibrio alginolyticus. And it seems to be that those Vibrios that fill in are more associated with pathology. We have genes that related to type 3 and type 6 secretion systems. These aren't virulence. These don't cause harm in it by themselves, but these are the mechanisms that bacteria use to invade host cells and then use those other virulence factors. These are things like Vibrio, cholera factor, cholera toxins, a whole suite of toxins. We're not even close to understanding, say, the mechanisms of pathology here. But all we know is that we get this shift in the functional capacity of the bacteria that are associated with oysters that we see during a mortality event. Another thing, and this is just building on some of this work, is another postdoc in my lab, Dr. Mark Krzyzewski. And what Mark's done, this is really neat. He's been developing these digital droplet PCR assays. And they're pretty cool. As scientists, they're really cool. As scientists, we use PCR to detect environmental DNA. This is a really common technique to detect or quantify the amount of DNA in a system. And you can associate the amount of DNA with, say, the amount of some target. DNA itself doesn't necessarily have to be a live target. It's just a fingerprint of the target, something that you're looking for. And this is what's used in like DNA barcoding and crime scenes and things like that. What's neat about the droplet PCR assays, and you're starting to see these used more in medical research, is that not only do you get the quantity of the environmental DNA, but you get some understanding of the sequence variation. You get some understanding of the sequence variation. Yeah, there we go. And so what Mark's been able to do is not just quantify the total amount of DNA that's present as we move into some of these oyster mortality events, but kind of a relative understanding of the community that's there. And what Mark has been seeing is that as we move into the mortality event, there's this kind of reshuffling of the Vibrio community. It's kind of got like a 3-day periodicity to it, both in the water around an oyster farm. So oysters are, the communities, bacterial communities within oysters are shuffling and changing with respect to virulence. But they're also changing in the water itself. And it's about 3 days. And what we think is happening here is that we have new niche space or new opportunities be created within oysters themselves as they move into a mortality event. And what that resident community is sloughed away with any oyster tissues in the water, and it gets replaced by some other community, only for the same thing to happen, say, 3 days later. So there's constant reshuffling. And that's what Mark is showing here in these data. The mortality event is that dashed line that you see there. Leading into the mortality event, we have these 3-day spikes in the water column Vibrios. The whole community is shifting. And what we're looking at the bottom, moving into a mortality event, is the total concentration of Vibrio DNA in oyster tissues. So leading into the mortality event, there's a little bit there, but not much. And then right before mortality, they really spike. And they shuffle. So one of these questions, we're starting to identify a characteristic pathology. I called it irritable bowel syndrome, associated with SUMS. We see this pathology consistently year after year. We're starting to see these microbial shifts emerge. We don't necessarily have a pathogen, but we do have these microbial communities that change and may be associated with pathogenicity. This question is like, why is this happening? We still don't know, but we're moving towards this. One of the things that we've done, this is some work funded by the North Carolina Department of Environmental Quality, also Sea Grant to an extent. We have a research hatchery at UNC Wilmington. Amy Wilbur runs that lab. What we've done is the majority of our industry imports seed from out of state, predominantly Virginia. We want to just ask this question, and some of you may have heard SUMS also referred to as triploid mortality. We want to ask this question of, is it the genetic origin or the genetics of the broodstock or ploidy itself that are contributing to what we're seeing with respect to SUMS? And so we've made paired lines where we've crossed Henry's commercially available diploids and tetraploids, and then say like a Virginia line and deployed it to North Carolina farm sites. But we've also made a North Carolina line. We have a handful of North Carolina diploid lines that we then also cross with a commercially available tetraploid from Virginia. We call that a North Carolina triploid, but it's not really a North Carolina triploid. It's like a hybrid type. And the results are really, well, striking. We deployed these to about, I think it was 8 field sites total. The best performing line in North Carolina, by far, were our North Carolina diploids. The worst performing line, by far, was the Virginia diploids. So it doesn't really seem to be a ploidy thing. The 2 triploid lines are like kind of intermediate. And so what this tells me is that the genetics is what matters here, probably more so than ploidy. Genetics is part of ploidy. there are only so many tetraploid lines that we can select from. And so genetics is inherent in ploidy. But it's the genetics itself that was most important. That's really associated with risks, or risk of mortality in sums. I want to just highlight, I'm going to stop talking in a minute, but I want to highlight just where some of this work is going. You can see on the top left of that, we have our HC diploid. That's Hewlett Creek diploid. This is our North Carolina diploid line. We did have one site where the mortality was really, really bad, and we lost a lot of those HC diploid lines. And I think it's important to note this. When the mortality is like really, really bad, genetics doesn't seem to matter. Everything dies. And I think what I think is happening there is it's related to a lot of that microbial turnover that we're seeing and the shift towards microbial species associated with oysters that are associated with like, a lot of these virulence factors in their genomes. And so there could be a lot of secondary pathology, and that can spread. And then it doesn't matter what your genetics are. But what we did with this deployment is in our North Carolina diploid lines that we saw overall perform really well, except at this one site where we had a lot of mortality. We took those survivors and spawned them. We also took their cousins that were deployed to a site that didn't see a lot of mortality and spawned them. So now we had our maybe resilient North Carolina diploid line, but now one that's gone through a SUMS event and has been selected to some extent. And then we deployed those, and the results are exactly as you say. Amy Wilbur did this. Here's a photo of Amy. Is that the North Carolina line that was selected through a SUMS event ended up performing better. So we're starting to see these incremental gains in performance with more lines that are going through SUMS. And this is where a lot of our work is going. We've now developed a handful of these candidate genetic lines where the broodstock have been exposed to a SUMS event across multiple sites, pull them together and spawn, and we made offspring, and I'm deploying them to sites that do and do not, or are thought to experience a SUMS event. And the hope here is to start to develop some idea of resilience. It does open up a handful of other things that we can do with just understanding some of the genes associated with the resilience and some of the processes, some of the epidemiological processes associated with resilience. So I'm going to stop here. Just want to give the overview of what we've learned is that we can call SUMS like an inflammatory disease. We see a lot of this inflammation. I say diverticulitis here, inflammation in the diverticula and the digestive organs. It isn't just a diverticula. We see a lot of this broad inflammation. Ploidy seems to be a secondary risk factor. It's the genetics that really seems to matter. Etiology is still unknown. We don't know what causes this. But there are some exciting things, including exciting things happening. I mean, the research labs of the people right here. Andrea is about to show some really cool work showing maybe viral or small bacterial etiology, which makes sense based on some of the things that I've seen in our deployments. I do want to highlight, we've seen this genetic component to it. And so what does that even mean? And some of the work that I'm really excited about is Bill is doing with Steve Roberts at the University of Washington. I think this has, as a pathologist, this has all the hallmarks of a metabolic disease. So really understanding metabolic stress in oysters and how that's contributing to susceptibility to SUMS. So I'm going to pass this to Andrea.
Can I steal a clicker? Thank you. All right, thanks, Tal. For y'all that don't know me, I'm Andrea Tarnecki. I'm an assistant extension professor and director of the Auburn Shellfish Lab. And so today I'm going to be talking about what we've seen from SUMS from a golf standpoint. If I can find the right button. Big green button means go. Okay, so a little bit of history. The first SUMS event that I know of was reported in 2016. I do want to say that I have only been at the Shellfish Lab since December of 2021, so I may have missed a few in this, but we have seen definitely a consistent reoccurrence of this since 2023. This is kind of a timeline going from March of 2023 through December of 2025, and I've got each state kind of on a different row, and the X's mark the SUMS-like events that I've heard about or have been reported to me from the different states. They're kind of sporadic, but what you can generally see is that they seem to be increasing each year, and that could just be because more people are talking to me about it. But I do want to say that we seem to see these hot spots between spring and fall, which isn't incredibly helpful. It just means that we've never had one reported in January. But we do kind of see peaks generally from March to May, and then often again in the fall between August and October. And through some of our experiments, we've accidentally captured some of these events. These are various deployments of research projects that had absolutely nothing to do with SUMS. We were testing different lines of oysters at different sites. But for each of these consecutive years, we caught one in 2023 in tetraploids at 2 different sites. We had them deployed at a Dauphin Island site and our Grand Bay Oyster Park. So we caught, again, that spring mortality event in tetraploids in 2023. We caught it in diploids and triploids in 2024 around the same time, and then again in diploids and triploids in 2025 around the same time. And one thing that I didn't include here because we got mortality data a little bit differently, we caught the fall of 2025 event in one of my grad student projects where we were supposed to be testing the effectiveness of bird deterrence. So most of those oysters died, and that was fun. But that's kind of what we see. We put these experiments out there and then accidentally catch a SUMS event. So there are some consistencies. They are kind of your characteristic SUMS events. They typically happen in market or near market size oysters. Oftentimes, these oysters are nearing a year old, often approaching their second summer of life. This is a very rapid onset of mortality, as you could see from the steep declines in survival on the previous graphs. And sometimes they can reach up to 100% in a few weeks. These are not associated with harmful algal blooms. They're not associated with parasitic dermo-infections, so we can rule those out for the most part. We do know that if you take oysters from a site that's experiencing mortality and move it to one that's not, they're still dying. We can't seem to save them by moving to a different location. What's been really interesting is that we're seeing this recurring theme of kind of a 1 to 2 month delay in die-offs. So what do I mean about that? Well, in 2025, one of the experiments I just showed you, we started seeing diploid mortalities in April. And the triploids looked great. We came back the next month to look at everything. Almost all the diploids were dead, and now the triploids were starting to die. So there was kind of a month gap in between those. At the same site, there's a commercial oyster farm, and 1 to 2 months after we started seeing mortalities, the commercial farmer started noting mortalities in their stock. So also, we moved oysters from an unaffected location to one that was seeing SUMS events, and it took 1 to 2 months before we started seeing dead oysters in that batch. And then an experiment I'm going to show you in a second, we started demonstrating these rapid mortality events in, it was about 40 to 45 days, so again, that 1 to 2 month period. So that's a really interesting recurring theme that we've got in mind that we're looking further into. But by far, there's a lot more inconsistencies than consistencies. So in 2023 and 2024, the first mortality events came reported from triploids, but in 2025, most of the mortality events that were reported and what we saw on our research farm, the diploids started dying first. So as Tal mentioned, ploidy does not seem to be the driving factor. We do not see consistent irregularities in water quality. So there were 2 events reported in Alabama where people reported a strange smell and a film on the water, which kind of passed quickly, which is disturbing in itself, but not reported in every mortality event. Also, 2 different mortality events, 2 oyster farmers reported high turbidity, but again, that's not consistent across these events. We see them in both primarily reports in domesticated or selected lines, but I do have to premise that by saying those are also the ones that we're checking the most often, so they're the ones more likely to see it in. But we do have controls in those that are a little closer to what you would have genetically for a wild stock, and when these mortality events hit, they hit that kind of wild line as well. Seasonality, most of the reports are in spring and fall when the temperatures are rapidly increasing or decreasing. But again, we're seeing it in summer and in winter where temperatures are consistently warm or consistently cooler. And because we're seeing it across basically the whole year, we're catching the rainy season and the drought season. So we're seeing it anywhere from 5 to 30 parts per thousand. So salinity doesn't seem to be the main effect. And then handling stress, there's anecdotal evidence that oysters that are getting desiccated or tumbled are being impacted harder, but again, that's not always the case. So another thing is, if you look at the oysters during one of these events, there's no consistent outward signs of that something is wrong. On the far left, this is a nice healthy oyster. It's got good color, the gills are rounded, the labial palps, which I don't have a pointer, but they look great. Going across from left to right, we see shriveled gills. We see gills that are like choppy with chunks taken out. We see shriveled labial palps, which is kind of in the bottom there. And then the one on the far right is really interesting. We had 2 oysters survive one of these mortality events. When you pop them open, they have almost no gills left. These oysters were alive, but they didn't have any gill tissue. So I don't think they were going to make it too much longer. But both of them that survived looked like that. So we see that on occasion, but again, it's not consistent. So in April to May of last year, I launched an experiment to see if we could replicate a SUMS-like event in healthy oysters. I could do just like 45 minutes on this study alone, so I'm going to condense it really as best I can. But basically, we took fresh dead oysters from a SUMS event, ground them up into a beautiful oyster milkshake, and then filtered it through different filter sizes. The goal of that was to isolate a fraction that would have, parasitic cells, which are the biggest cell types, one that had bacterial cells, which are kind of intermediate, and then one that had viral particles, which would have been the smallest size. We took those fractions and injected them into the adductor muscle of apparently healthy oysters. I say apparently because we don't know what causes it, and as I said, when you pop them open, most of the time they look healthy, so apparently healthy oysters. And then we tracked the mortality for those for 50 days. I'm not going to go through all the results, but I just wanted to show you. We also had some oysters that were injected with sterile seawater and kind of the oyster milkshake from healthy oysters, just to show that if you inject oysters with those 2 things, they don't die. So they're not dying as a result of the injection process, and that healthy oysters don't have the same things that fresh dead ones do. So the black line is the seawater, and the orange is the healthy oyster, and this is mortality over time. They both ended with 75% or higher survival. But for the dead viral fraction, the virus fraction that came from dead oysters, they had a little drop off between 20 and 25 days, nearing that month mark. But then 41 days, we started seeing a substantial crash, and within 7 days, we lost them all. That's kind of characteristic of a SUMS event. Again, it's at that 1 to 2 month period. So this tells us that we can potentially replicate SUMS-like events in healthy oysters using the viral component. Again, 1 to 2 months. And some of them that we popped open did have some of those strange gill and labial palp appearances that I mentioned earlier. So it doesn't rule out the role of viruses in these events, but I really want to stress this because I don't want y'all going around saying this is caused by a virus. There are other things present in that fraction. There's proteins, there's potential toxins, microbacteria, and other things that we haven't identified. So it's not just viruses in that fraction. And we have only done this study once, so we haven't replicated it. So it could have just been an artifact of that study. But it's something really interesting that we're going to continue to follow. So we're going to continue those injection studies. We have 2 farms in Alabama that are participating in the Oyster South SUMS project, where we're going to be monitoring oysters over time at 2 locations. And then we have conditionally funded Mississippi, Alabama Sea Grant projects. One is going to use hydrographic models and machine learning to look for patterns in these mortalities, so maybe identifying some of these stressors that we don't see just by looking at reports. That's led by Dr. Anna Linhaus at Auburn. I'm not a modeler, so I can't answer too many questions there, but we're going to help her collect data for that. The second one, we're going to track bacteria and viruses like Tal was talking about to see if we see similar shifts like they do in North Carolina, and if we can identify some early warning signs that this event might be coming. And then we're going to work with a commercial farmer to look at submerged water graders to reduce handling stress and hopefully improve survival. So what do we know? Unfortunately, like Tal said, we don't know the cause. Multiple stressors are probably involved. We've ruled out a lot of things on their own. Viruses or other small particles may potentially play a role in that. But I just want to end by saying that Every report and observation that y'all have is helpful to us. It can feed into our modeling. We can ask questions and kind of narrow things down and maybe try and find some pattern that we haven't seen before. So if you have a mortality event, please reach out to me or some of the other extension agents, talk to them, and we can gather that data and put it in and hopefully make some progress on this issue. So thank you very much. I'm going to hand it over to Ryan. We'll hold questions till the end. Thanks, Robert.
Okay. Hi, everybody. Can you hear me okay? Yep, good. Okay, so I need to advance. So I'm Ryan Carnegie. I'm here from the Virginia Institute of Marine Science. First, I'd like to compliment Tal and Andrea for the level of granular detail they provided. I mean, this is an enigmatic disease that we're trying to understand, but the stuff that they presented is careful experimental work that is yielding insights that will get us closer to answers and to solutions. So that's all good. What I'm going to present over the next few minutes, and fairly briefly, is sort of a higher level perspective on our impressions from our work in Virginia, which extends back to 2012 and something like 200 plus industry cases we've received from Maine to Texas, in which we've sort of done pathology to try to understand, what the pattern and type of pathology is that's associated with this condition. And I'll present some of the things that I'm thinking about for future directions and things that I think you all could be engaged with. So, advancing the slide. So, what is sums from our impressions? So, and these are typical oysters that we receive in pathology cases to our lab. So this is this sudden mortality that we tend to see in oysters from intensive aquaculture, from the springtime into the early summer especially, sometimes extending into the middle part of the summer. Oysters may be dying in very significant numbers with mortality above 50, sometimes approaching 100%. But typically the oysters are arriving in very good condition, not like we would see in a typical dermo disease case. And yeah, we tend to think that this is because of the timing of the mortality, around the time of peak reproduction, that somehow mixed up in this as a cause is the stress of reproduction. Even triploids, which are mostly sterile, are trying to produce some amount of gonad. It doesn't develop to mature gametes that they're releasing, but diploids and triploids both are producing gonad that represents a metabolic burden that they're compelled to maintain at a tough time of year because the water temperatures are high, and especially after the spring bloom is past, the food quantity and quality in the systems may be sort of subpar. It may be below what they need. So these are animals that have been bred to sprint, and they may be basically encountering challenging environmental conditions that are tipping them over the edge into significant levels of mortality. So that's sort of the vision we have for this. And again, as has been mentioned, we don't see obvious pathogens among the major known pathogens associated with this disease in our systems. Though I am entirely willing to believe that there could be viral or bacterial engagement in the process. We have over the years tended to focus on protozoan pathogens, which are most of the major pathogens of oysters in the east and along the Gulf. And we've had a blind spot with regard to bacteria and viruses that the work by Tal and Andrea will be beginning to fill. And so this is sort of the overall kind of model that we see for how all this might be working, that whatever is going on, it probably represents some interaction between the genetics of the animals in production, something in our husbandry practices, which could be fouling control, which could be density, there could be other factors. But again, as you have been hearing, something in a more challenging marine environment, which may be warmer, which may be increasingly hypoxic, especially in the center of crowded oyster bags, as Bill and his students have demonstrated. So this is kind of what we believe is kind of coming together. My focus, as I tend to think about this, is often on the genetics. We've been very successful in oyster breeding over the years, dating back to the 1960s, breeding for fast growth, breeding for MSX resistance, breeding for dermo resistance, which really tends to be the primary focus of the breeding programs, dermo resistance and fast growth. Are we sort of creating, as we focus so single-mindedly on growth rate and dermo resistance, is it possible that our genetic constructs that we're growing on these farms actually are less resilient in the face of sort of environmental stressors? I don't have an answer to that question, but that's something that we're increasingly talking about. We're doing experimental work at VIMS, funded by the USDA, to start getting at some of these questions. And we really want the geneticists to start thinking about this. Is there a cost to fast growth and dermo resistance that we may need to deal with in terms of general resilience? Waffle House, the Waffle House Index. And you all may have heard a bit about our application of this to oyster production. This is this index created by a FEMA administrator in the region, looking at waffle houses distributed throughout the South, whether they're open fully or partially open with a limited menu or closed, as an indicator of the broad geographical impact of natural disasters like hurricanes in the Southeast region. So, we've thought about this with regard to shellfish production and SUMS in particular. Like, can we sort of engage industry at broad scales to get insight into how things are going? So I'm really excited about the various projects that are going on this front. So you've heard reference of the Oyster South-led Farms project, which is working with a number of the farms, extension agents, and scientists in the room here presently. So that's sort of widening the circle a little bit, working with a number of farms broadly throughout the region. But we're thinking, and Bill, he may be able to comment on this after, we're looking to try to even broaden this, more and more, to try to gain feedback from, dozens to hundreds, if possible, of farms on how they're doing, what they're seeing, the timing of problems as they emerge, so we could start connecting that to the modeling that we may be able to do, looking at data sets from the buoys, the satellites, the stream flow gauges, and so on, to put together observations of impacted performance at the farm level throughout the region, to put that together with things that are going on and meteorologically or oceanographically, to try to make sense of drivers at the broadest scale. And if we can do that, if we can start to understand what might be going on behind SUMS in terms of the drivers, we might be able to eventually get to where we can make predictions of when farmers in different regions are going to start to see animals under stress. So that's at the broadest scale. I think we need to be interested also in what's going on at the smallest scales. And this is just a photomicrograph of some histopathology. These are oyster gills. We generally see oysters coming into the lab that are in great shape, that have been submitted to us amidst these SUMS events. The most notable disease sign that we see associated with these problems is gill pathology. And so we viewed this as sort of a signature or a sign or a key symptom of SUMS, because we tend to see more of this sign in aquaculture populations that are under stress. But we're viewing this increasingly as a potential cause of the problem. These oysters that where the gills are affected, there may be hemocyte infiltration, the oyster blood cells flooding the organ. If that's happening, it could be impacting the ability of the animal to push blood through its gills and thus to oxygenate its deeper tissues, which could contribute to tipping an oyster over to mortality at a challenging time of year. If the gills are ravaged, as we could see in this image, they've lost all of their external epithelial cells. You just see the skeletal elements of the gills left. If they've lost that epithelium, if they've lost those cilia, they're unable to feed. So that translates to nutritional stress that could be compounding oxygen stress. So I think as we move forward, thinking about the pathology that we see and how it might connect to things at even the small scales in terms of physics and chemistry happening on an oyster farm and inside oyster bags is going to be very constructive to better understanding SUMS. And I will leave it at that.
What I will do, sorry, guys. What I was going to do is Colin Elmers had to go home. He has great information as a student with Tal. So what we're going to do is we're going to ask Colin to record his talk, and we're going to share that with Oyster South so that you can get that full information instead of trying to keep you here and rush through it. All right? So apologies to Colin for not being here and not giving his talk, but we will make that available to attendees with a link after the symposium. John, can you go to the last slide? Because the only thing I want to add to this is Ryan mentioned the Waffle House Index, and it's confidential, so I'm not asking anyone to raise hands who participated, but we got a great set of data from that of where people were reporting concerns with mortality across the U.S. And we were up to, I think, over 90 farms by the end of the year. We would love to start that again, but I don't want to bother you with that request without you signing up again. So thank you, Andrea. The last slide here, if you can show that, that's on the right-hand side is showing you some of the data that we got, and then you can sign up for this year's. As a grower, you can sign up. We would love, the intake form maybe takes 10 minutes, but the weekly, I know that sounds like a lot, the weekly form takes, on average, under 30 seconds to fill out. You're just asked, what is your level of concern with your oysters that are going to market this year with the level of mortality? Green, yellow, red. Click. If you want to put a comment in, you can. But otherwise, you can be done that fast, and we'll start to assemble this, and we'll share the results with you. It has been, you would ask, why is this important? I know you want to get to solutions. One way to get to solutions is to show that this problem is significant. If you've had SUMS, you know it's significant. If you haven't had this, or if you're a panelist on a review for proposals that we put in, and you don't have data like this, they can say, I don't think that's that important. I think it's because of husbandry, et cetera, et cetera. It's too easily dismissed. So we need these kind of data to learn about this, but this also supports us trying to solve this. So please sign up for that. I know we're at lunch, but this is a super important issue for growers. So I think we actually start again at 1:15. So I just want to hold people for a couple more minutes. Do people have questions that they'd like to ask the panelists? I know, Robert? Yeah. Okay, the question was, has there been a stop point? I don't know if, Ryan, you want to talk to that, or Tal?
So we have definitely gotten reports of mortality that would fit the pattern of SUMS from Maine. A couple of years ago, some of the regulators had mentioned that, yeah, they think they're seeing the same thing up there. We've gotten some samples for pathology that suggest, yeah, it's the same thing. There's unusual mortality that's elevated, very high, but no obvious, and early to midsummer, but with no obvious engagement of the major pathogens. So.
Right, I'll just add that it's not isolated to Virginica. There's mortality, and it kind of fits the bill of SUMS.
Oh, so Navy Cove Oyster Company's on the South Shore Mobile Bay, and 2 summers ago, we experienced what we considered a SUMS event, 40% loss. So, we send up oysters to Ryan's lab, and here comes the report. No known agent. Multiple stressors, husbandry, question mark. And we oyster farmers, the only thing we can really do about this is take a hard look at what we're doing with these oysters. And what we did is we just changed things. So the oysters spend very little time out of the water during grading. And as we started approaching the warmer water temperature months, we started harvesting out of the middle of our oyster grow cages. So we're leaving the center open to get more water flow through the cage. And I think what's very important is you reduce stress, because that's all you can do. So take a hard look at your husbandry, because I think it's very important, and that's the reason why he's making a point.
While I walk this over to the next person I think, yeah, Mark, right there. I just want to add, I think it is critical to say that husbandry might be one of the ways to ameliorate or mitigate this, but I have seen farms operated very, very well where we've seen these. So I do not want to say that this is caused by farmers, because I think that's going down a path where it's easy for then people to dismiss this, or for insurance claims to say it's your fault. I think husbandry can help mitigate the other things that are driving this.
Hi. Hi, so Mark Casey, Delaware farmer. We've been hit by this several times. The survey that we do weekly, I would say, is great. Thank you. As your pictures showed, I know I'm about 6 weeks late on reporting it because I can't tell by looking at the outside of an oyster. So think about how you could get this program to more of a shuckers view and how we can educate shuckers to say that it's starting so we can start to head it off. The baby change husbandry, I think, is an important piece we all need to work together with. And then my second question to you up there is, I know that the pea crabs die first. So if you're looking at these things and you're looking at the pea crabs, they're dead 2 or 3 weeks before the oyster's dead. And there's some synergy there. So if you can help us figure that out, that would be great too.
All right, I think we'll go to Skid for the last question.
Real quick, one of the things I try to advise people is to have some pets. And by that I mean some oysters that are set aside and getting the best treatment possible in a bag at low density, that's never fouled, that you're checking every week. And, to say that it's husbandry puts it on the grower, I don't necessarily think that's true because it could be just a change in the environmental conditions that is saying that the husbandry that's worked forever is not working anymore. But my question is to Tal. It looks to me like there's a genetic component. We have this East Coast Breeding Consortium. We have this SNP chip that's supposedly got, 60,000 single nucleotide polymorphism mutations identified. How can we use that to identify what's different about your strains that are not dying and the ones that are? And is that work being done, please?
Yeah, Bob, absolutely. What we've started doing is we're tagging oysters before deployment and not genotyping everything, but genotyping a big chunk and then seeing the survivors, and we have the genotypes because they're all tagged. And that's going back to the SNP chip. And right now we're doing the whole genome association studies, but it's exactly that. What are some of the markers associated with resilience? And how do we build that into the existing breeding program?
All right, on that, I almost killed myself on the comfort monitor. I just want to point out, I know there's some great work. If you're having these issues, please reach out to either these folks, your extension agents. I know Leslie's done a lot of stuff in Florida. Like, there are a lot of people that want to help with this. Oyster South is leading, in addition to being associated with the Waffle House Index, there is currently a project where the seed are going in the water. I think after the symposium, we're all going back, and we have 20 collaborating farms from Virginia to Texas where we're putting diploids and triploids in the water through the year, monitoring environmental parameters in the bag. So we'll be able to report on that as well. But that's the type of work that we're hoping to get done, and we want to help. And so we need you to collaborate with us and let us know what's going on.